Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. Good evening all of you. Yes. Welcome back to Unacademy NEET English. Audio video is absolutely normal and I just got it checked. Yes. Good evening everyone. So how are you all? Excited to start the new journey for NEET 2027, which is going to be a different journey because of so many reasons, and we know that already. So are we all excited? Give me a thumbs up, send me all the green hearts. How many of you are excited for preparing for 2027? This is going to be a huge challenge, and it's going to not only raise our confidence level, but we are actually going to establish a new history here in the field of NEET. Yes, we'll be the first batch who is going to take CBT, and we are going to prepare with the bestest of our abilities. You have all the facilities here which will help you prepare for that. Good evening. And I am going to, wow, thank you so much everyone. And before I start with today's session, guys, first of all, to all my future doctors, a very happy Doctor's Day. How many of you know that it is Doctor's Day today, and it is such an auspicious day that we are starting our Phoenix 2.0 biology batch today. I know it has already started yesterday, but the biology batch is starting today. This is the first live class on YT. Rest of the classes will follow on the Learners app as well. But today's class is for everyone. Good evening, all of you. Good evening. Yes, guys. Let me quickly check once. Quickly like, subscribe and share. Everybody. I really don't know what has happened. I think now it is absolutely normal, right? All good now? It's all good now, right? Perfect. So yes, so all those of you, good evening. Class starting soon. Yes, it is fine. Never mind small glitches. You know, I have come to realize whenever there's a glitch in the beginning of my work, now my whole work goes very well. So I actually look forward to a small glitch now in the beginning of my work. Anyways, all right. All right, guys. So yes, once again, a very important piece of information. Yes, you can access all the batches, right? Test series and upcoming course, that is Phoenix 2.0 batch. The plus batch you can get at 4,699 only till tonight. You have to use the code TR Live. And with this, guys, I am going to start with today's session. So guys, today we are going to start with the first chapter. The first chapter here for Phoenix 2.0 batch is, we are going to start with the very basic structure of life. And what is the basic structure of life? That is cell. We are going to start with this chapter. Give me a thumbs up that you're all set with new notebooks ready for making the class notes. Class notes are going to be very important. Now, before guys, I start with this chapter, let me tell you what are going to be the rules of these classes. So all my plus students know that already who have joined the batch, but those of you who are watching me for the first time and who want to know how the plus class actually goes on, so you must know how do we conduct our class. See, first of all, we are going to discuss the whole chapter. So there is discussion of the whole chapter in detail. First, there will be discussion of the chapter in detail, right? So when we say detail, that means I will be describing the chapter, all the contents, right? And you will be making the class notes. Of course, at the end of the class notes, you do get a PDF, but the most important notes are always handwritten notes that are done by you. In the class, I'll be giving you quick points also, right? We'll be discussing some PYQs, the questions that have already come in the exam, right? After the completion of the chapter, I will be reading NCERT in the class. I have done that already. We'll continue the practice. Okay. Other than NCERT, we'll be discussing NCERT Exemplar. Now, all the students who have already attended few classes with me in the plus batch, batch, please let me know. Have we discussed NCERT in the class, or have we discussed NCERT exemplar or not? Quickly let me know. Yes. Everyone, all of you tell threes. Dr. NEET journey, what is this? Why are you spamming? Yes, there will be 3 sessions, guys. This is just a beginning. Let us set the pace, right? There will be 3 batches as you have been informed. Don't worry about anything. We know that we have to complete the syllabus of Phoenix 2.0 by February. And after February also we have to do 2 revisions before your NEET exam, NEET 2027. Okay, so don't worry about that. Everything is planned. Okay. Now there are proper, you have already got a schedule also that which chapter will be allotted how many lectures. So everything is planned according to February. Okay, and if you will cooperate, we can also finish it little faster and little jaldi. Is that clear? So NCERT exemplar. After NCERT exemplar, guys, we'll be discussing the PYQs and we'll be highlighting the PYQs in NCERT also. So if you want that you should get all these PYQs marked NCERT, you should join my batch also, sorry, Telegram channel also, Tina Ma'am Biology. I'll be posting all the DPPs and all on this channel, Telegram channel. Is that clear? Now with this, I am going to start. This is going to be the way of our lectures. And apart from that, you know that you can always ask your doubts in the classes. Doubt asking is very, very important. So I highly appreciate those students who will ask their doubts on daily basis. There are separate doubt taking sessions also. There are mentors available, but you can ask your doubts directly in the live class also. Now, all those of you who are already aware of this, give me a thumbs up and I can start with the chapter. Yes, guys. Please let me know. Any book you should buy for NCERT NEET 2027 biology preparation apart from NCERT? No. You don't have to buy any other book. Okay, let me clarify this thing also. What are the books that you have to follow? The books for NEET 2027. There is only one book, that is NCERT, and the sister of NCERT, the sister book, which is NCERT exemplar, which has lots of questions. And those who have already solved these questions with me, they know how important this book is, how good questions there are there in exemplar. And there are some small topics which are not given in textbook NCERT, but the questions have been asked. So exemplar also gives you an insight which extra topic has to be done. Not many, hardly 1 or 2 topics are there in each chapter, but then we do get a direction how extra has to be done. Otherwise, we just have to restrict to NCERT. And now if you have gone through the NEET, re-NEET exam, you know that some unexpected questions are also asked. The formula based numericals have been asked this year. So we are going to prepare in all the best possible ways. So there is just one book that you have to prepare from. The other, you don't need any other book. You need more question practice. And where do you get all these questions? All these questions, there are so many, thousands of questions are given in Unacademy Learners app already. You don't have to hunt for any other book. No other book, guys, no other book. The more books you will accumulate at this stage, there are higher chances of missing the chance of clearing NEET. Is that clear? Now that is a final advice and a verdict also, and a warning also. Advice, verdict, warning, all the 3. Do not buy any extra book. Solve as many questions with us in the special classes, in the live classes, right? And on the Learners app, there's a huge question bank, topic wise, unit wise, chapter wise, the whole syllabus wise. Are you getting it? Now, is there any question that you want to ask? Any other question that anybody wants to ask? If you have any other practice book, yes, you can solve. But now, see, why am I saying now you should not buy any other book? There's a a big reason. The pattern has changed. It is CBT now. You are asking me for some particular books which are otherwise very famous in the market, right? Very good books, no complaints. But now I have been advising all my students to do questions online. Get in the habit that you solve these questions just by looking at the screen. You will not be given pen and paper where you can encircle the keywords, right? There are higher chances of you making mistakes when you are reading the question online, yes or no. How many of you agree with me? The speed is going to decrease. So you have to work on all these challenges by solving questions online. And if you want better practice, please go for CBT. Without CBT, it is going to be, you know the answer. Are you getting it? We don't have to prepare for the syllabus only. We not only have to prepare for the syllabus, we have to prepare for a new exam format also. And guys, I have already made a small video, hardly 10-minute video, where I have discussed what are going to be the challenges for you for NEET 2027. So please watch that once again, okay? And watch that and write down few pointers that you have to take care of during your whole preparation now. Now, please tell me, shall we start with the session? Rakshana, no, I would not suggest you to buy any books now. Guys, please start a new habit. Please get into this habit of solving questions online. Stop, give up on this habit of solving questions on pen and paper mode because you don't have the leverage of marking the keywords now. Please do that. You can't mark the keywords. Now that is going to be a huge disadvantage, but you have to work on that now. So how will you work? Try to solve more and more questions directly from the app, directly online. Work and time management is going to be a very important factor this year. Very important factor. So we'll be working on these things. We'll be developing on this foundation. Is that clear? You know how to study, right? We will be giving you some more tricks, more ways how to learn it in an easier way. But now the new challenge is not the syllabus. The new challenge is CBT. So first of all, see, it is always better to know the challenge. Know your bimari, know your disease, know your challenge. Then you work on it. Accept it, change, adapt, right? Adaptation is the key to survival. Simple as that. And now on that note, all my future doctors, let's start with the chapter that is cell. Right? Is that clear? I actually hate doing this way. Honestly, even me too. I'm also very old school. I love having a book in my hand and underlining things. But now, see, if you have to survive, you have to adapt to new ways, right? Now guys, let's start with the first chapter of Phoenix 2.0 batch, that is cell. Now cell, what do we already know about cell? Cell is the basic structural and functional unit of life. Yes or no? Structural and functional unit of life. When we say this, what do we mean by this statement? When we say that it is a structural and the functional unit of life, what do we mean by that? We mean that all the living structures in our body, all the living structures are always made up of cell. Anything which is not made up of cell cannot be regarded as living. Is that clear? Now, functional unit means all the functions are actually performed by the cell. You see your lungs involved in gaseous exchange. Who is involved? It is basically a cell. There are cells which are making that organ which is involved in a particular function. So all the functions, digestion, why do we have to breathe? Because a cell is producing some gases. Cell needs oxygen. Cell is producing CO2. So we need that gaseous exchange. Why do we have to digest? Because we have to digest, we have to break down the food to make it small enough to reach up to the cell. Why do we excrete? Because the cells are producing the waste. Are you getting that? So everything is occurring at the cellular level. Where is digestion occurring? Your cells produce lysosomes. There are enzymes, right? Lysosomes are the bodies which contain enzymes which are involved in digestion. Are you getting that? So everything is actually occurring at the cellular level. Your brain, all this electrical activity, who's conducting it? Neuron, the longest cell of the body. So cell is a basic structural and functional unit of life. So it is the cell which makes something living. So without cell, life cannot exist. So there is a line given in your NCERT, anything lesser than cell is not living, is not living. That is why guys, that is why virus is not considered a living till it is not within the host, right? So virus is not considered living outside the host body, not considered living outside the host body. Please tell me these points are clear. Is that clear? So anything less than cell is not living. So it means the first thing which decides whether this particular structure can be called living or not, that is cell. Is that clear? So that is cell. So that is why when we study what is living, then cellular organization is one of the criteria. Are you getting it? So anything less than cell. So if there are cells present, it is called living. If the body is not made up of cells, it is known as non-living. Also known as animate or inanimate or non-animate. Please tell me these points are clear, right? Now, we understand that sometimes cells can group to form tissues, tissues will form organs, organs can form organ systems, and then the organism, like in our case. But that is not always the case. Now, there can be two situations. Cell may have an independent existence, right? So, cell, if may have independent existence. That means only one cell is able to carry out all the life processes and doesn't depend on any other cell. So, independent existence, then in that case, it is known as a unicellular organism. Then the single cell, when single cell has independent existence, does not depend on any other cell and can survive alone, can behave as an organism, this is known as unicellular organism. For example, amoeba, paramecium, all the protozoans that we talk about, right? But if there are so many cells in the body, multiple cells present in the body, then in that case, the organism is known as multicellular organism. Of course, we are also the example of that. Please tell me these points are clear to you. Is this point clear to everyone? Yes, guys. Is this point clear to everyone? Anyone with any doubt till here? Very easy, right? Now let's go back in little history here. Let's talk about the background. How did we discover the cell? Background, or let's say history. So we know that discovery of cell was made by Robert Hooke. Everybody knows that? We have been learning this since, I think, 6th, 7th standard, right? So discovery of cell was done by Robert Hooke. Now Robert Hooke basically discovered the dead cell. Which dead cell did he discover? Cork cells. So cork cells look like this, like a honeycomb-like structure it has. It has this honeycomb-like structure. So when he took a slice of cork and observed it under microscope, he observed that it has these compartment-like structures, right? And there was nothing in between, in the center. So they were nothing but empty. These were empty compartments. As observed by Robert Hooke. Is that clear? So Robert Hooke said these are empty compartments. Compartments can also be known as cells, and that is how cells got discovered. So remember, Robert Hooke discovered the Dead cell. Which cell did he discover? Cork cell. How did it look like? Honeycomb-like structure. It had honeycomb-like structure. Honeycomb means beehive. Is this clear? But now, if we talk about the living cell, the discovery of first living cell, that was made by, first living cell was discovered by Leeuwenhoek. Discovered by, living cell was discovered by Anton von Leeuwenhoek. Is this clear? Anton von Leeuwenhoek. Please tell me these points are clear to all. So this was Robert Hooke, and the living cell was discovered by Anton von Leeuwenhoek. Is that clear? Now, what are the cells discovered by Anton von Leeuwenhoek? So he observed spermatozoa, human sperm, right? RBCs, then bacteria. He observed all these, and also some protozoans, unicellular eukaryotic organisms. So he observed these cells for the first time. So these are the cells discovered by him. Then we have one more scientist here who discovered the nucleus. So the nucleus was discovered by, who? Robert Brown. So that was Robert Hooke. Nucleus was discovered by Robert Brown. Please tell me these points are clear to you all. Yes, guys. Are these points clear to everyone? This is Robert Brown. Please tell me these points are clear to everyone. Yes, guys. Any doubt till here? Any doubt till here? In the end of the session, guys, we will be discussing the PYQs. Yes, is it done? Right. So, clear to everyone? Now, the details of the cell will be discovered as the details of the cell got discovered as our technology advanced, right? So first we had a very basic microscope. Then Leeuwenhoek was able to see all these structures by his own self-made microscope, and gradually as the technology advanced, we found electron microscope. We invented electron microscope. We were able to see the cell in detail, right? So we have so many different we found so many different details and intricacies of the cell. Even we got to understand that the cell can be classified under prokaryotic type and eukaryotic type. So all these intricate things about cell we are going to understand in this chapter. Please tell me these points are clear. Then another thing, we have cell theory. We have cell theory. Now see, cell theory was proposed by two people. It was proposed by two people. Both of them were German. One was a German botanist. See, nationalities are important, right? And other was a German zoologist. They were not friends. They were not making any collaborative study. They did their studies independently, right? So what is the name of German botanist here? The name of the German botanist is Matthias Schleiden. Sorry, that was. So it was Matthias Schleiden. While the German zoologist is Theodor Schwann. Is that clear? So these are the two scientists who gave the cell theory. Is that clear? Now, German botanist, that was in 1838. What did he study? Since he is a botanist, so he is going to study the plants, and he observed so many plants, and he proposed that all plants are made up of cells. Is that clear? All plants are made up of cells. That was his proposition. Is this point clear? Is this point clear? Then What about German zoologist? So he was making his independent research. Theodor Schwann in the year 1839. See the years. First came Schleiden, then the Schwann. Now, Schwann in 1839, what did he propose? So since he is a zoologist, he observed many animal cells and he observed that all animals are also made up of cells. Are also made up of cells, right? Now, because Schwann came in 1839, no doubt he was making an independent study, but of course he was aware of what Schleiden has been doing. So he had also studied what Schleiden had observed, right? So he said that all animals are definitely made up of cells, but he said that all plants, see, this he said that cell wall, which is present in the plant cell wall, which is a characteristic of plant cell, is not found in animals. So the second proposition was that cell wall is unique to plant cell. Why did Schwann give this thing? Why did Schwann propose this point about plants? Because when he was studying, when he was confirming things about the animals, he did not saw any cell wall. So he said the cell wall which is in plants is unique to plants. It is not found in animal cell. Please tell me these points are clear. Please tell me these points are clear, right? And also he said that all these cells have an outer membrane known as plasma membrane. Please tell me you're clear with these points, and you must know them separately. What are the propositions by Schleiden? What are the propositions by Schwann? Please tell me, guys, that these points are clear to all. Everyone. Is that clear? So it means basically overall the cell theory was formulated by two people, a botanist and a zoologist. Is that clear? But now neither of the two is telling us how did the first cell come, or how do the new cells come. So nobody was able, neither Schwann nor Schleiden was able to decide that, or to propose that, or discover that, or inform us anything about the formation of new cells. So gradually, as the time advances, came Rudolf Virchow. So came Rudolf Virchow. Okay. So now Rudolf Virchow came in 1855. 1855, and he gave us this idea that cells come from the pre-existing cells. So cells, new cells, come from the division of pre-existing cells. Is this clear? How do these cells divide? This we'll understand in the chapter of cell cycle and division, a beautiful chapter. Now, new cells come from division of pre-existing cells. But how did Rudolf Virchow said that in his own scientific language? See, he did not say these scientists, they don't say these things in a very easy way. Have you ever seen a doctor talking? They'll always, use their medical terms and all those hi-fi things, right? So, how did Rudolf Virchow say? Rudolf Virchow said the same thing as omnis cellula e cellulae. Omnis cellula e cellulae. So, what does omnis cellula e cellulae means? It means that cells come from pre-existing cells. Please tell me. So, this is how the cell theory got modified. So after 1839 came when the cell theory was proposed, Rudolf Virchow modified the cell theory. So who modified the cell theory? Rudolf Virchow. So when you solve questions, read the statement very carefully. Sometimes it is written, cell theory is given by Schwann and Schleiden and modified by Rudolf Virchow, right? Absolutely. If they say it was given by Rudolf Virchow and modified by Schwann and Schleiden, will this statement be right or wrong? Please let me know. Can't see the English batch. It doesn't appear under the please check if I have been enrolled. Sarcastic, please let me know. Please join the batch tomorrow, or message me on the Telegram, or contact the team also. Okay, contact the team. What is the number to contact the team? 8585858585. So this is how you can contact the team and get your resolution done, get your problem resolved. Wrong, right? So what is the statement? See, so can we say that Schwann and Schleiden, see, this is the question, Schwann and Schleiden formulated the cell theory, and it was, which was modified by Rudolf Virchow. Yes. Now, if they have to complicate the question even more, how can they complicate? They can add the years here and switch the years. Schwann and Schleiden formulated the cell theory in 1855, which was modified by Rudolf Virchow in 1839. Will the statement be right? Now, will the statement be right? No. So you should also know the years. This was done in 1839 and modified by Virchow in 1855. So 4 different statements can be easily made. Yes or no? Is this clear? All right. Done. So we have studied about cell theory. Clear to all? So what is the ultimate thing about cell theory? What can we say about the cell theory? That cell theory, what does cell theory state? That all the living beings, be it plants or be it animals, they are made up of cells. Can we say that? That is the first conclusion. And second, all the cells arise from pre-existing cells. So these are the 2 statements. So what is the ultimate thing? All living beings are made up of cells and their products. Products means it can be some of their secretions also, which contribute towards the formation of living being, right? So this is one. And second, all cells arise from the pre-existing cells. Please tell me these points are super clear to you. So this is the overall cell theory, which was formulated by Schleiden and Schwann but modified by Rudolf Virchow. Are these points clear? These are the key points. Of cell theory. Please tell me these points are clear to you all. Yes, please. Give me a thumbs up, guys. Are you making the notes along? This class will go on till 8:30, guys. Class will go on till 8:30. 1.5 hour class, first class. Otherwise, my classes will be for 2 hours. This is just the first class, right? Now, is this point clear? Everyone, quickly let me know. Are you making the notes? Are you making the notes alongside? Right? All right. Now, moving on to the next part of the chapter, that is overview of the cell. Let's have an overview of the cell. Overview of cell. Now, what do we understand by overview? A rough idea, a rough sketch, a general sketch. Is that clear? Let's have an overview of the cell. Now, if you observe any cell, you want to study a plant cell, you can take an onion root cell because it's easy to study, easy to obtain. Or you can just simply take a leaf of onion, peel it, the inner transparent sheet, right? You can observe that also easily. For animal cell, the easy option is to scrape off your inner lining of the cheek. You know, you take a slide, right? And when you do it like this on the inner side of the cheek, so there'll be some saliva on that cheek, saliva on the slide. So that saliva basically contains your cheek cells also. You make a smear of it, observe under the microscope, and you can clearly see how cells look like. So when you see a typical cell, be it inner lining of the cheek or a cell of a onion, right, onion peel, what is the general structure that you will observe? Now see, the general thing that you will observe, for example, onion cell or human cheek cell, whatever, or you can take any other cell also, human cheek cell. What do you observe? Now see, what will we observe? We will observe three basic things. We'll observe three basic things. Plasma membrane, right? And a central nucleus. Plasma membrane and a central nucleus. Okay? Now, if I say, I'll specifically tell you, if I take onion cell, right? If I take onion cell, how will the onion cells look like? Onion cells will look like more like this. Okay. So these are the plasma membranes. So this is the plasma membrane. Now plasma membrane is common to both plant cell and an animal cell. Is that clear? And we'll see a dense structure which is called nucleus. Right? And surrounding the nucleus, we'll see this semi-fluid thing which is known as cytoplasm. Now, since we are talking about the onion cell, which is a plant cell, then apart from plasma membrane, outer side of the plasma membrane will show the presence of another layer which is called cell wall. This is the overview. This is what we observe in a typical cell. Is that clear? This is what we see in a typical cell. Yes or no? Now, both the cells that I am studying here, just to have an overview, I'm giving you an overview, the basic structure, right? So, every cell will have a plasma membrane. Most of the cells will have a nucleus also. There are few examples where nucleus are absent. Now, why that is absent, we'll come across that reason also. Every cell will have cytoplasm. As far as it is living, it will have a cytoplasm. And now, this is unique to plants only. So, cell wall is not seen in animal cell. Schwann has already authenticated that idea. Is that clear to all? Right? So, you will see the cell wall in onion cell, but not seen in human cheek cell. Are these points clear to all? Please tell me. Are these points clear to everyone? Now, that is why. Now, what does NCERT write? NCERT says plasma membrane is a delimiting membrane. Delimiting membrane. Delimiting membrane means it will form the outer limit. Is that clear? So, according to NCERT, NCERT has clearly written, see, the cells of human cheek has an outer membrane as a delimiting structure. This is the delimiting structure, membrane or a delimiting structure. So, it forms the outer boundary. Where? In human cheek cell, animal cell. Not the same thing said about plants, because plants will still have an outer cell wall. Is this point clear to all? Please tell me. Then, about nucleus, NCERT is saying that nucleus is a dense, round structure. Dense, round structure. Please tell me. And it is membrane bound. Dense, round structure and membrane bound. We'll see the details of all these structures, why and how it is membrane bound, how is the membrane like. We'll see it all. Then about cytoplasm. Yes, guys. About the cytoplasm. Cytoplasm is a semi-fluid matrix. Semi-fluid matrix. It's not fluid, it is semi-fluid matrix. And this is the site where all the cellular activities will take place. So, NCERT will write down this point that cytoplasm is the arena for cellular activities. What is arena? Arena is the area where fighters fight, right? Where wrestlers fight. So, this is the arena, this is the ground, this is the playground, this is the fighting ground for all the cellular activities, cytoplasm. If cytoplasm ceases to exist, we will see that gradually there are certain cells who will lose their cytoplasm. If the cytoplasm will be lost, the cell will become dead. Now that is also a part of their life, because loss of cytoplasm is also contributing towards some function that we will study eventually. But initially, a living cell will always have cytoplasm. If the cell will lose cytoplasm, it will die. Dead cells are also part of certain living body. See, understand, a living body, a living organism can also have dead cells which are showing some function, some specific roles in the body. But overall, we are living, right? But yes, then dead cells also have an important role. We'll discover them one by one. Is this point clear? Now, apart from these structures, there are some other structures like endoplasmic reticulum, mitochondria, Golgi bodies, right, etc., etc. All these structures are membrane-bound structures. These are membrane-bound structures. Is this clear? Right? Now, overview. There are membrane-bound structures. Then there is this structure called, this structure known as ribosomes. Now, ribosomes are always membraneless. They will never have a membrane around it. It is always membraneless. And what is the function of ribosomes? We all know they are the protein factories. So, protein synthesis is the function. Please tell me all these points are clear to everyone. Delimiting, Jivesh. Delimiting means creating a limit, that this is the limit of the plasma. This is the limit of the cell. Beyond this, the cell is not existing. This is the outer boundary. Delimiting membrane means outer boundary. Outermost boundary. Is that clear? Right, guys? Now, see, now understand, there are certain structures which will always be present. As I said, plasma membrane will always be present. Cytoplasm will always be present in a living cell. Now, nucleus may or may not be present. Cell wall may or may not be present and may be made up of different chemicals. Then membrane-bound structures may or may not be present. Ribosomes are present, but then these ribosomes can also be of different types. Now, what I am trying to tell you that there are two types of cells based on these differences. Two types of cells. See, two types of cells. One is the prokaryotic cell and another is the eukaryotic cell. So guys, those of you who are in class 11, maybe, is there anyone who is from class 11 who is aiming for NEET 2028 and is attending this session? Is there anyone? Please let me know. Please let me know, guys. Is there anyone who is aiming for NEET 2028 and still here? No? Yes? If you are there, if anybody from NEET 2028 is here, the content is exactly the same. Just that for NEET 2027, we have to complete both 11th and 12th in one year, and for 28 we have a lot of time and we can go like step by step, right? but the content remains the same, the speed remains the same. I am not missing on anything. I'm starting from zero, and as I say, I'll take it to the next level, NEET level. Is that clear? So even if you have never studied this chapter, you can still watch this session. But basically, this is an insight into our plus classes, how we conduct. Now listen, prokaryotic cell. See, so prokaryotic cell and eukaryotic cell. First of all, what does it mean? How do you break the word? Pro means primitive. Karyon means nucleus, basically. U means true, karyon means nucleus. So true nucleus. So cells containing true nucleus, and these are the cells containing primitive nucleus. So what does primitive nucleus look like? So basically we say that they don't have a typical nucleus. Their genetic material, DNA, is lying freely in cytoplasm. Now whatever I am writing down here, theoretically, I'll be describing in the diagrammatic form also just after this, okay? But first, just understand the basic differences. So genetic material is lying freely in the cytoplasm. Why it is lying freely in the cytoplasm? Because it is not membrane bound. Because it is not membrane bound. Not membrane bound. Is that clear? So there is no membrane which will limit the presence of genetic material. It is lying just randomly in the cytoplasm. Okay. Now the region where this genetic material is present, so that region is known as nucleoid and not nucleus. So nucleus is absent here. Nucleus is absent. Instead, the region as where the genetic material extends in the cytoplasm, that region is known as, that region is known as nucleoid. So we don't have a nucleus in prokaryotic cell, we have a nucleoid. Is that clear? It's not only that there is no membrane around the genetic material, there is no membrane bound organelle present here. So all the membrane bound organelles like endoplasmic reticulum, mitochondria, GB, Golgi body, right, etc. All these are absent. These are just the basic differences. The details of prokaryotic cell I'll discuss in the diagram only. Is that clear? Now, what about the eukaryotic cell? Here, the genetic material is bound within a structure known as a nuclear membrane. And that whole region is, that structure is called nucleus. The genetic material lies within within the nucleus. That is, it is membrane bound. It is membrane bound. And it is not only the genetic material which is present within the membrane bound structure, rather there are other membrane bound structures also present. Other membrane bound structures also present. Is this clear? What are the membrane bound structures? You know that ER, GB, mitochondria and others also, we'll talk about it. Ribosomes, I told you, they will always be present, but again of different types. So ribosomes in here, in both eukaryotic and prokaryotic, ribosomes are present. Ribosomes are present. Tick. Tick means present. But here they are of smaller size and we call them 70S. Now what is S? What does 70? How does 70 come out? This value comes out to be. So we'll discover it all gradually. Here the ribosomes are bigger in size and we mark them as 80S. But having said this, eukaryotic cells will also retain some prokaryotic features. Okay, eukaryotic cells will retain some prokaryotic features, so they will have 80S as well as 70S ribosomes. So where are they present? We'll discuss it. Please let me know, guys, are these points clear to all? Drop or class 12 student. Ma'am, the screen is too bright. Can you continue the class? Bhavita, Let's see. I still don't know about the YouTube plan. I'll let you know about the YouTube plan, but definitely we have started the proper classes for NEET 2027 in a structured, in a well proctored way for 2027. Please tell me these points are clear. Okay? All right. Everybody done with this? Now there are more differences, and I'll be again coming on this topic, on this difference between prokaryotic and eukaryotic in a detailed manner after I have discussed each one of them in a lot of detail. Is that clear? This is just the overview. Please understand, this is just the overview of cells going on. Now tell me, can I start with the details? Yes, please. Can we start with the details? Yes or no? All right. So let's do one thing. We'll start with the details in the next class. I'll still give you some more idea about the cells here. See, now the cells, we are still talking about the overview. So the cells will, coming up onto the cell shape and structure. See, the shape and the size, shapes, structure and size, right? So the shape and size of the cell, that depends a lot on its function. Hashtag, it depends a lot on its function. See, I always give this example. Whenever you are designing your room, you know, there is always a thought process behind it. If you are a student, you will always require a study table, then a bed, a smaller bed around, right? If you're designing your house, you know, you design things as per the functionality. You will not keep a crockery cabinet in your study room or in your bedroom. You will not keep refrigerator in your bedroom, right? Where do you require these two things? In your kitchen. So the function, so the arrangement of the structure depends upon the function it has, right? Similarly, the cell shape will also depend upon the function it has to do. Now, I have just a short while ago, I said there are certain cells which will lose cytoplasm. Again, that depends upon the function. There are certain cells which will lose their nucleus. Again, that depends upon the functionality of the cell. But now we are talking about a typical cell. So let's talk more about it. See, first of all, the smallest living cell here, smallest living cell, we'll talk about certain things. The smallest living cell, then largest living cell, longest living cell. What are these? Smallest living cell is mycoplasma, right? Largest cell is the ostrich egg. So if anybody asks you, have you ever seen a cell with the naked eye? Anybody who has seen a cell with the naked eye? Don't write ostrich egg. I am sure you have not seen an ostrich egg. Hardly anybody would have visited Australia and got this chance. Do not write ostrich egg here. But still, my question remains: have or have you not seen a cell with the naked eye? Please let me know in the chat. How many of you say that you have not seen a cell? You are saying no. I have seen. You have seen. I am saying with the naked eye. Okay? Don't try to tell me that you have seen through a microscope. That is not a naked eye. That is through microscope. With a naked eye. Has everybody seen a cell? Yes? You saw it in your school? Puvan? Have you not seen an anda? Anda ka fanda? Anda, everybody, egg. Whether you are vegetarian or not, we have all seen eggs. Egg is a cell. Every murgi ka anda is a cell. Yes or no? So we all have seen a cell with the naked eye. Okay. But the largest cell is the ostrich cell. Longest cell is the, what is the longest cell? It is a neuron. Or you can say a nerve cell. Is that clear? You can say a nerve cell. Right? Now the size. Now the smallest cell, which is a mycoplasma, the smallest cell, the size is only 0.3 micron meter. Is that clear? 0.3 micron meter. So this is about the size. If I have to give you some more list about the sizes, see bacteria, we can say bacteria, the size of bacteria will be between 3 to 5 micron meter. That depends on which bacteria we are talking about. Is that clear to all? Then there are some other sizes of, let's say, human RBC. Human RBC, the size is around 7 micron meter. Is that clear? Human RBC, 7 micron meter. Clear to all? Now, a typical eukaryotic cell. What is the size of typical eukaryotic cell? The size is between 10 to 20 micron meter. Yes, please. A typical eukaryotic cell size is between 10 to 20 micron meter. Now, please note down that in the theoretical part of NCERT, they are writing the size of bacteria as 3 to 5 micron meter, while just on the next page, I'll be reading NCERT also, but just on the next page in a diagram, they are writing the bacterial cell, the size of the bacterial cell lies between 1 to 2 micron meter. So both the facts are absolutely right, because it entirely depends upon the type of bacteria that you are observing. Bacteria can be as small as this or bigger than that. Is that clear to all? Then there is PPLO, pleuropneumonia-like organisms. These PPLOs, the size is about 0.1 micron meter. I hope we all know that 1 micron is 10 raised to power minus 6 meter. Clear to everyone? Now, virus is not living as long as it is outside the host body, but still we'll talk about virus. So the size of virus is actually even smaller than mycoplasma, which is 0.02 to 0.2 micron meter. Is that clear? But here we are talking about the smallest living cell. Virus is not always considered living. Is that clear to all? So note down these sizes. These sizes are important. You have got this question in the match the following form already. So we have discussed the size here. Let's talk about the shape. Now, talking about the shape of the cell. Shape of the cell, as I said, again depends upon the function. It again depends upon the function. Is that clear? Now see, for example, shape of the cell will again depend upon the function. So, cells can be disc shape, discoidal, or they can be polygonal, or they can be rounded, round, or they can be columnar, right? They can be cuboidal or flat, right? They can be flattened also. They can be very elongated cells also. So all these shapes will actually depend upon the function they perform. For example, discoidal cells. This is the shape of RBC. Why this RBC acquires a discoidal shape? Because discoidal shape seems to have a very large surface area. Is this clear? And RBC has to get involved in gaseous exchange. So because RBC is involved in gaseous exchange, discoidal shape will have large surface area. This has large surface area. So it is an advantage for gaseous exchange. And on top of it, we'll understand gradually that RBC will lose all the structures to accommodate more hemoglobin. Are you getting that? Right? So understood the significance of shape here? Polygonal shape for tight fitting of the cells, right? When cells have to be tightly packed like this. Round, there will be some intercellular spaces here. How do columnar look like? This is how columnar cells look like. Then cuboidal cells are more squarish like this, right? Then flattened cells, if the cells are flat, they are important for diffusion. Diffusion of gases will not occur across any other cell but here. So if you want a diffusion membrane, you need these cells. If you want the cells to squeeze out of the smallest spaces also, what would be the shape? It will have amoeboid shape. Amoeboid shape means no fixed shape. It can change, it can squeeze through. And WBCs have such shape. Are you getting that? They can squeeze out elongated structures. So your neuron has to carry distances, carry messages to the long distances. So neurons will have such shape. Your muscle cell has to contract. So muscle cell will have spindle shape. Are you getting that? So the shape depends a lot on the function that it performs. Please tell me that point is clear. Please tell me these points are clear to all. Yes, guys. Clear to everyone? How is nucleoid different from nucleus? Rakshana, I will be discussing details of nucleoid, but in next class, okay? Is that point clear to everyone? So that was the overview. Now, next that we have to start with is prokaryotic cell. But before I start this, guys, we'll start with the details of prokaryotic cell. Before I start the prokaryotic cell, I want to remind all of you once again. So this is the first class of Phoenix batch, guys, and today we are going to end this offer where you can get the plus batch for only 4,699, and you want to buy at this price. This offer is ending tonight. You have to use this code TRLIFE. That is my code. Okay. Now, more important, if some of you have already taken this batch, I'm sure there are some students who are already a part of Nexus and Phoenix batches. Those of you who have already taken this batch, I am reminding all of you again. Again, it is a warning, it's a verdict, and please understand, 2027 preparation is not just about syllabus completion or understanding the syllabus. It is about understanding and preparing ourselves for a new exam format. Please do watch the challenges that I have discussed in another video. Okay. It is important that you know what we are going to face this year or in the NEET 2027. So you start preparing at the right time. So you have to get the batch. If you have taken the batch already, you have to get it upgraded to CBT. Link is given in the description box. Without CBT preparation, just preparing the syllabus will not help this year, guys. So shift your mode, adapt. Okay, now, is that clear to everyone? So we have discussed, I have told you how our scheme is going to be. I'll be uploading today's notes on this Telegram channel, and DPPs are also uploaded here. All the short notes after every chapter, the point-wise short notes that I have prepared by me, that are also shared on the Telegram channel. I have already done that for the previous chapters. I'll do that for this also at the end. Now, books that you have to follow, only this. Where are you going to practice the questions from? From the Unacademy Learners app. You don't have to hunt for any questions. There are books available though, but now I'm telling you, you are going to prepare for CBT. So just pen and paper preparation would not serve your purpose. Start, give yourself that challenge. Be habitual of reading question online and solving it. Then we have started with this chapter. We have discussed the background, some discovery, cell theory, and the overview of the cell. The two types of cell, their sizes and their shapes. Please tell me these points are clear to everyone. Yes, guys. Yes, please. Please let me know if there is any doubt till now. Anyone with any doubt till now? Is it all clear? Is it all clear? Right. All right then. All right. Now let's start with perfect. All clear? Ma'am, Schwann was a, both were German. Ruchir, if you're using the previous NCRT, the old NCRT, it was written British zoologist, but it has already been rectified. Both are German now. So NCRT suddenly found out the new nationality of Theodore Schwann. How can we challenge their nationality here? And NCRT has said he's German, he's German. Earlier NCRT thought he was British, he was British. Okay. Chalo. Which link do you want? How many questions will come from this chapter? So by the way, if you want to see the breakdown of this chapter, so overall guys, there were 9 to 10 questions, 9 questions I think from this whole unit. So 2 to 3 questions from this chapter every year. See, I'll tell you exactly. 2026 we got the exam that got cancelled, 4 questions from this chapter. 25 had how many questions? 5 questions again from this chapter. So 3 to 5 questions from this chapter are coming every year. Is that clear? Now let's talk about the prokaryotic cell. Yes guys, let's talk about the prokaryotic cell. Give me 1 second. Okay, now that's a problem. Anyways, yes guys, let's talk about a prokaryotic cell. What does the prokaryotic cell consist of? Number 1, like every other cell, see, there will be cell envelope. There will be cell envelope, cytoplasm, then the genetic material present in this region known as nucleoid. Okay. Now, the cell envelope, the cell envelope, cytoplasm and nucleoid, right? Now, cell envelope consists of 3 layers. We have the outermost glycocalyx, then we have cell wall, and we have plasma membrane. These 3 layers collectively are known as cell envelope. Now, understand, glycocalyx may or may not be present. Positive means may be present, negative means may not be present, right? Cell wall again may or may not be present. Is that clear? Plasma membrane is always positive. Clear to all? We'll discuss their details. First, just understand what are the different structures. Cytoplasm. Cytoplasm, we have read that it is a semi-fluid nature. Whether it is a prokaryotic or a eukaryotic cell, cytoplasm will always remain a semi-fluid matrix. Now, what does the cytoplasm consist of? Cytoplasm will contain some organelles. There is only one organelle present, which is called ribosome. Ribosomes. There are no other membrane-bound organelles present, right? Ribosomes, and that too of 70S type. Then there are some inclusion bodies. Now, what are inclusion bodies? We'll see that. And of course, there is a matrix where cellular activities are occurring and where these structures are also present. That is known as cytosol. Okay, or you just call it matrix or cytosol. So all these things collectively form cytoplasm, right? And then we have nucleoid. So nucleoid is the region where genetic material is present. Genetic material is present. And this genetic material is known as, see here, this genetic material will be known as prochromosome. We don't use the term chromosome in case of prokaryotes. Why? We'll understand. Here we'll call it prochromosome, or we can call it genophore. Is that clear? We can call it genophore. Now, apart from nucleoid, which has the main genetic material, there can be an extra chromosomal genetic material also, and that extra chromosomal genetic material is present in the form of plasmid. It is present in the form of plasmid. What is it, this plasmid? Plasmid is the genetic material other than this nucleoid, other than this main genetic material. So it is extra chromosomal genetic material. We will talk more about each structure in detail in the next class. So have you understood the basic categorization of the structures present in the prokaryotic cell? Yes, guys. Also, now see, please understand, this plasma membrane will form certain structures. This plasma membrane will form certain structures, like it can form mesosome, right? It can form chromatophores. It can form the spillae or fimbriae. So what is the function of each? We'll discover it gradually. Now, understand one last point. These mesosome and chromatophores, they are formed by the invagination of plasma membrane. Now, what is the function of each? That is a part of detailed analysis of all these structures. While pillae and fimbriae, they are formed by the evagination, that means outgrowth. Invagination, ingrowth, evagination, outgrowth. Is that clear? Just giving you a hint, pillae and fimbriae are meant for attachment, not for movement. If the bacterial cell or the prokaryotic cell has to be motile, then it must possess a structure known as flagella. Again, flagella may or may not be present, but this flagella will be involved in movement. Is this clear? For movement. So it may or may not be present. Again, please tell me all these points are clear to you. Yes, please. Now, when we say prokaryotic cell, it is supposed to have this basic structure. There will be variations. Why mesosomes? Why chromatophores? Which will have mesosome? What does it do? All a part of detailed study. Now, let me tell you the organisms which have prokaryotic cell, whose cell is prokaryotic in nature. Now, the examples, write down the examples here. Examples are, number 1, bacteria. Second example, mycoplasma. Third, we can say PPLO. And fourth is this cyanobacteria or blue-green algae. So these are all examples. Please tell me these points are clear to all. Yes, guys. Anyone, you guys all from the batch? Yeah. If yes, how did the test happen? Yes. Tests are regular. Gram positive, gram negative. Yeah. We'll talk about gram positive, gram negative, but that is a part of details. Okay. All right, guys, please tell me if you are thorough with this part. Quickly let me know. Are you thorough with this part? Anyone with any doubt? And give me a thumbs up if you are totally thorough, there is no doubt at all. Yes, please. And then I'll start with the basic of cell envelope here. Yes, please. I'll start with the basics of cell envelope here. All thorough? Okay. Now, I will just start with cell envelope. Let's see how far can we go here. Now, first of all, I have said That cell envelope is made up of three layers: glycocalyx, cell wall, and plasma membrane. And also we have another question also here. So outside to inside, if you have to arrange the structure from outside to inside, so this is outer, this is innermost. So glycocalyx, if present, is the outermost layer. Inner to that is cell wall, and innermost will be plasma membrane. Let me tell you how. So see, this is the plasma membrane. Okay. Now, outer to plasma membrane, we have outer to plasma membrane, we have cell wall. This is a cell wall. Okay, now please understand, there is no gap between them. I am just keeping a little gap to keep their identity clear. Okay, so let's label it. Yeah, so this one is the plasma membrane. This one is the plasma membrane, right? Which will always be present. Outer to that, we have cell wall. Now there can be difference in cell wall, okay? And the outermost is the glycocalyx. This is all glycocalyx. Is that clear? This is glycocalyx. Clear? Glycocalyx, then cell wall, and inner to that is the plasma membrane. Now I have already mentioned the fact glycocalyx may or may not be present. Now first of all, glyco word, so it contains saccharides, it contains sugars, right? Now see, glycocalyx may or may not be present. What is the function of glycocalyx first of all? So bacteria which has glycocalyx, now this glycocalyx is going to help the bacteria to hide. It is like the invisibility cloak of Harry Potter. Okay, so Harry Potter used to wear that invisibility cloak and nobody could see where he's going. Similarly, if the bacteria has this glycocalyx on it, the WBCs, the immune system of the host organism, cannot see this bacteria. So suddenly the bacteria becomes invisible to our immune system. Are you understanding? So glycocalyx may or may not be present. So there are two strains, guys. There are two strains. We'll come to this when we read genetics, okay? But I'm telling you here itself. So glycocalyx, if it is present, then the bacteria will be known as smooth. It will have a very smooth layer around it. It is smooth. We call it S strain. So it is known as S strain. Clear? If the glycocalyx is absent, it is known as rough. So such bacteria is known as R strain. Is that clear? So the same bacteria can have two strains, R strain as well as S strain. Let's say it is Streptococcus pneumonia, the bacteria which causes pneumonia. Now all the Streptococcus pneumonia will not cause disease. Okay? There will be Streptococcus pneumonia which has glycocalyx, while the other form will not have glycocalyx. Is that clear? So the form which has glycocalyx is known as S strain. Which has glycocalyx will be invisible to our immune system. So therefore, what the bacteria which is invisible to our immune system will be able to hide itself and cause disease. So S strain is always disease causing. So it is always disease causing. What do you call it? Virulent. So S strains are virulent. Is that clear? So the same bacteria can be of S strain form or it can be R strain. R strain, rough, there is no glycocalyx, negative. What will happen? It will be visible to our immune system and our immune system is going to kill it. So if the immune system is able to see this bacteria, it is going to destroy it. So R strain will not be able to cause disease. So cannot cause disease. Cannot cause disease. Is this point? Therefore, these are known as non-virulent. So R strain is always non-virulent. virulent. Please tell me these points are clear. Right? One more thing. Now this glycocalyx can be loose, and if it is loose, it is known as slimy layer. If it is loose, we call it slimy layer. If it is hard, if it is tough, then the same glycocalyx will be known as capsule. Is that clear? It will be slimy. It can be slimy if loose. It will be known as capsule if it becomes hard. What is the function of glycocalyx? I just told you, it is the invisibility cloak. Hashtag helps the bacteria to hide from the host immune system. Host's immune system. Please tell me these points are clear to everyone. Yes, guys. Please tell me these points are clear to everyone. Anyone with any doubt? Anyone with any doubt? Is that clear? So guys, I think I'll stop here. There is probably another session aligned for you. So we have started with the cell envelope, and I have just talked about the first layer, outermost layer of cell envelope. I have just talked about first layer of cell envelope. So this is the outer one, this is in middle, and this is inner. If all the three are present, this is the order. Is that clear? All right, guys. So that is all for today. Once again, before going, I am reminding you, today is the last day for the offer, guys. So make sure that you use this code TR Live to get this offer, and all of you have to upgrade for the CBT preparation. So this is Phoenix 2.0. This was the first lecture, and rest of the classes will continue on Unacademy Learners app. I hope you liked this session. Once again, a very happy Doctor's Day to all the future doctors, and make sure that you do leave your comments. If there is any query, do let me know, right? I always read your comments, and use this power of comments in a positive way. Let us know whether you liked it or what are the challenges that you are facing. Do leave your comment after this session, and I will be uploading all the notes here on this Telegram channel, so make sure that you join this. Thank you so much. I'll see you tomorrow on Unacademy Learners app, Phoenix 2.0, tomorrow 7:15 once again. Bye guys, and all the very best.